Cells are the fundamental units of life, forming the building blocks of all living organisms. Despite this shared role, not all cells are created equal. They can be broadly classified into two categories: prokaryotic and eukaryotic cells. To truly understand the complexity of life, we need to compare and contrast these cell types and explore the scientific explanations for their differing levels of complexity. Prokaryotic cells are the simplest form of life. These cells lack a nucleus, a defining characteristic that sets them apart from eukaryotic cells. Instead of housing their genetic material in a membrane-bound nucleus, prokaryotes keep their DNA in a nucleoid region, a dense area within the cytoplasm. Prokaryotic cells are generally small, typically ranging from 1 to 10 micrometers in size. Their internal structure is relatively simple, with few specialized organelles. For example, prokaryotes possess ribosomes which are responsible for protein synthesis, but these ribosomes are smaller and less complex than those found in eukaryotic cells. Prokaryotic cells also have a cell membrane and, in many cases, a rigid cell wall that provides additional protection and shape. Some prokaryotes, like bacteria, may have specialized structures such as flagella or pili to aid in movement or adhesion. Eukaryotic cells, on the other hand, are significantly more complex. They are usually larger, ranging from 10 to 100 micrometers, and they contain a nucleus where their genetic material is stored within a double membrane. This nucleus is a hallmark of eukaryotic cells and reflects their advanced level of organization. Beyond the nucleus, eukaryotic cells are characterized by their abundance of membrane-bound organelles. For instance, they have mitochondria, the powerhouse of the cell, which generate energy through cellular respiration. Plant eukaryotic cells also contain chloroplasts, enabling them to carry out photosynthesis. The endoplasmic reticulum and Golgi apparatus are additional examples of organelles that contribute to protein synthesis, folding, and transport. These organelles compartmentalize cellular functions, allowing eukaryotic cells to perform complex tasks efficiently. This compartmentalization is a major advantage and a key difference from prokaryotic cells. When comparing the two types of cells, their structural differences point to significant variations in their functions and capabilities. Prokaryotic cells, due to their simplicity, are limited in their ability to perform specialized functions. They thrive in a wide variety of environments and reproduce rapidly through binary fission, a process of simple cell division. Their simplicity is a strength in terms of adaptability and survival in extreme conditions. Conversely, eukaryotic cells, with their intricate internal organization, support the development of multicellular organisms and enable the specialization of cells into tissues and organs. This complexity allows eukaryotic cells to sustain life forms with higher functions, such as humans and other animals. To understand how cellular complexity evolved, scientists have developed various explanations. One widely accepted theory is the endosymbiotic theory, which provides an evolutionary explanation for the origin of eukaryotic cells. According to this theory, eukaryotic cells arose from a symbiotic relationship between primitive prokaryotic cells. The theory suggests that an ancestral eukaryote engulfed smaller prokaryotic cells, which then became integrated into the host cell as organelles. For example, mitochondria and chloroplasts are thought to have originated from free-living prokaryotes that were incorporated into early eukaryotic cells. This theory is supported by evidence such as the double membranes of these organelles and their own DNA, which is similar to that of prokaryotes. Another scientific explanation for cellular complexity is the role of gene duplication and mutation over time. As genetic material duplicates and mutates, new genes can evolve to perform novel functions. This process allows for the development of specialized proteins and structures, increasing the overall complexity of eukaryotic cells. Over billions of years, these genetic changes have enabled the evolution of the diverse range of life forms we see today. Despite their differences, both prokaryotic and eukaryotic cells play essential roles in the web of life. Prokaryotic cells, such as bacteria, are crucial for processes like nitrogen fixation and decomposition, which maintain ecosystem balance. Eukaryotic cells, forming the basis of plants, animals, and fungi, drive the development of intricate biological systems. By studying these cells and their complexity, scientists can continue to uncover the fascinating story of life's evolution and interconnectedness. In summary, Prokaryotic and eukaryotic cells differ in complexity, size, and structure. Prokaryotes are simple, efficient, and adaptable, while eukaryotes are complex, specialized, and capable of supporting multicellular organisms. Scientific explanations such as the endosymbiotic theory and genetic evolution shed light on how cellular complexity has developed over time. Together, these two cell types provide a complete picture of the diversity and ingenuity of life. Their differences and shared characteristics highlight the intricate interplay of simplicity and complexity that drives the natural world.